And so we'll just start with a little bit about CHINTA and what is the philosophy that we are trying to follow here and why. This is like we are trying to do research in the neuroscience domain, but across spectrum of neuroscience, right? From bench to bedside, which is really talking about the basic or the fundamental aspects of neuroscience, all the way to trying to find novel therapeutic strategies for complex disorders. Now, one of the things in 21st century, one of the problems, can I change using this? Let me just try this. All right. So one of the challenges in 21st century is the problem of the brain, right? These neurological disorders that span across lifespan, all the way from some of these really early on developmental disorders, such as autism that you might have heard of, during your lifespan, such as stress, depression, at all varying levels of depression or anxiety. And towards the end, you have this neurodegeneration, right? So you have these neurodegenerative disorders like Parkinson's or Alzheimer's. So this is pretty much, I think, the problem of 21st century is the problem of the mind. We have, gotten handle of some of these other peripheral disorders, like, if you talk about bacterial infections, if you talk about cardiac problems, we have a good control. We can control the heart rate as precisely to like a minute, 1 beat per minute, right? But these, we still pretty much are not able to really have any, these are, most of them are still incurable, right? They are still, we only do things in a rehabilitative manner where we have interventions to just try to rehabilitate some of the symptoms and rescue some of the things. The reason is we really don't understand much about the mechanism of why it's happening. We don't understand the targets or the mechanisms to target if you want to develop a drug, right? And so one of the things that CHINTA is to really ask questions across biological scales, which is what's required to really understand a particular disease or disorder at all these scales. And so we, like I said, we work at all different spectrum, all the way from genes to behavior. And I'll give you guys a bit of an overview of all the different groups that are working in these different scales. But the reason why we need to understand each of these, or understand the disorder in each of these different scales, is because that's when we'll be able to really find effective therapeutic strategies. If we don't understand the genetics, if we don't understand what's happening at the level of synapses, if you only assume what we see in the behavior is the phenotype, then we'll not be able to really find that effective strategy. And so understanding that any kind of neurological disorder, or even understanding fundamental neuroscience across scales, becomes really key to studying neuroscience. And this is our core philosophy at CHINTA. Another problem in neuroscience, and why this is so important in 2025 to do this in India, to have these dedicated neuroscience institute, is because a lot of the times, or a lot of these kind of studies across scales that we talk about, or we get this inspiration from the West, right? This has been done a lot more extensively in the West with really good infrastructure and infrastructure which is specific for neuroscience research. But Indians are different, right? We don't really have a systematic way to really look at any neurological disorder, be it Alzheimer's or Parkinson's. We might have heard about them in the Indian context, right? So not in an Indian specific manner. So we have different genes. We all know that, right? We are not similar to Caucasians. We have different lifestyles and diets, and we have different metabolic and physiological makeup, right? And we also definitely have different socio-economic context. And if we look into all these different factors, they all contribute to any of your neurological disorders that you can think of, right? And so to really understand neuroscience for Indian specific population, we do need to study it in the Indian specific context. And like I said, a lot of the drugs that we are using to treat any of our disorders are really based on studies done in Caucasians. And so we need both from the discovery side all the way to the drug delivery side, we need to do studies in an Indian specific manner. Now, we also know that if you just look into the degeneration side, the aging population in India is actually increasing very significantly. As we compare from 1950 to 2050, we have almost five-fold increase in the aging population. And so to keep the quality of life similar, we need to really cope up with a lot of the degeneration that really happens with age, and to understand that, again, in an Indian specific context. Now, how do we study this? Like I was talking about different scales. Oh, this is just the prevalence of some of the degenerative disorders. We have almost a doubling of Parkinson's from 1999 to 2000. And then we have Alzheimer's and other dementia. Again, there is a tremendous increase in the burden over the last 3 decades. Now, what is the way forward? Like, how do we study it, right? I'm talking about really studying it in the Indian context, and really take example from the West and try to do cross scales. But what are the challenges? What really is the way forward? Right now, the tools that we use to look into some of these complex disorders are actually kind of primitive, kind of blunt. We really try to understand these disorders in a snapshot, or in a very crude manner. For example, we try to compare a healthy brain from a diseased brain, or from in a post-mortem brain tissue. So this is just telling you about the endpoint of that disease, right? We can't really tell the progression. Even if you want to tell the progression, we use structural MRI, where we look into the different structures of the brain. We try to understand or compare the volume of different brain regions, how that is changing, and comparing that with the healthy individuals. And we have functional MRI, which is not really used very extensively in India, but that tells you about the activity of the brain regions and how the different activity of different regions get associated during a particular complex behavior. So we can do these different behaviors and compare them with healthy and diseased individuals. But we still don't have the understanding or the resolution to really look at either the individual cells or the synapses or the specific mechanisms that get affected during any of these neurological disorders. So the brain is a lot more complex than just looking into these regions, right? Now we are in 2025. We should have a way to look at individual cells, right? Or not just cells, but even the changes in the connections, or what specific protein gets activated that leads to a lot of these neuronal functions. And we need a high resolution analysis of cells and molecules. And that is the reason, because we don't have that resolution, we still don't have an effective therapeutic strategy, right? Just by looking at these brain regions, we can get a sense or get an idea of some gross anomalies that might be seen in these diseases or disorders. But then what? What do we do after that, right? We need a better resolution to target something specific. We can't just, like, back in 1950s, we can't just remove a hippocampus and expect that the epilepsy is going to get better, right? We know now that hippocampus is important for so many different diseases, or it's important for so many different functions. Right? So we need a better resolution. And now at CHINTA, we are trying to really match these scales, or really map out from molecules all the way to the behavior. So we still do a lot of those. We use the blunt tools, like what Shona has pointed. This is his slide, by the way. So what he has said, like blunt tools, we still use that, but we now take one step forward and get this resolution that we really want to understand the mechanisms a bit better. So we go at the level of neurons, we go at the level of synapses, even in the patient brain cells or patient brain samples, and try to do some functional analysis of how do these connections, how do these circuits change, using many different cutting edge tools, which is, I think, the way to go forward. And the unique aspect of doing this here is to really look into it in an Indian context, right? So I'll just give you guys an example of that. All right. So at CHINTA, like I said, we do research at all a different spectrum. So I'll give you an example of one Indian specific disease that we are working on, or disorder that we're working on across spectrum. This disease you might have heard about is called spinocerebellar ataxia. 12. Have you guys heard about it? It's a ataxia, which is a movement disorder. So the not animal, but the humans, they're not able to walk, or they're not able to walk properly. Their gait is abnormal. This is an early onset ataxia. But in addition to that, they also have cognitive problems. Okay. And now this is a phenotype. This SCA12 specific mutation occurs only in the Marwari community of the Eastern India, okay, for whatever reason. But this is prevalent, or it's predominantly found in the Marwari community here in this West Bengal, Kolkata region. And so we took it up as a challenge, and we were like, okay, this is an interesting neurological problem. Let's just try to see, try to understand this across scales, like I said. And so we have a stem cell group, which is a big group that's working here. What they are doing is they take patient samples and they convert the blood cells into the stem cells. These are induced pluripotent stem cells. And then they can convert these iPSCs into neurons, cortical neurons, or dopaminergic neurons, or serotonergic neurons. This takes forever. This takes 6 months to a year. So this is not an easy technique. But once you have these neurons, which are mature neurons, of course, they can also have a co-culture of astrocytes and non-neuronal cells. So they can look at it in a population-like level. And then we have almost like a patient dish, or patient brain in a dish, right? So we can then do a lot of different experiments. The experiments that we are now doing, so this is, again, the stem cell part. We do a lot of these molecular and biochemical assays, compare them with the wild type or their control siblings. So people who have SCA12 mutation, they have brothers or sisters who don't have. So we take them as control. And then we take their bloods and convert their stem cells also into neurons, right? So that those will be our controls. So that's at the stem cell part. But then we can take these cells and we can also do look at their electrical properties. How do they communicate with the other neurons? We can do electrophysiology recordings and understand their synaptic properties, because a lot of the times in these neurological disorders or even developmental disorders, you have the communication that is affected. Okay. And so now we move up a little bit. So we have understood the genetic aspects. We are trying to understand the molecular aspects, understanding what kind of proteins get affected between disease and controls. Then we move up. We now get into the synaptic stage, where we're trying to understand synaptic physiology, how that is affected. So we do that using patch clamp electrophysiology. My group is involved in doing that aspect of research for SCA12. To also understand these synapses and correlate with behavior, we are also developing transgenic zebrafish lines. So we want to make animal models so that we can really use or do these mutations in an animal model and study it in zebrafish and see what kind of behavioral phenotypes you can see, what kind of protein changes we can see. It's not just a patient-specific mutation, it's also observed in zebrafish. And then we go further up. So I took a top bottom to up approach, but we go further up. We can even go and look at the patient's behavior, right? So from the patient that we took the samples, we can do a lot of different types of cognitive and behavioral assessments. We can do their brain scans, and we can try to extract a lot of information. So then we bring in the computational approaches that we can do to extract more information, see if we can make a predictive model to longitudinally see the degeneration based on some brain scans. And for that, we have a computational group. And so we have this across scale for the same disease. We are trying to use the different tools to ask the same question, like how did this, like what is really happening in these brains? And so this really brings us, or this is a lot more efficient to bring us one step closer to even finding a therapeutic option, right? Almost going for something like a personalized medicine. So that's one example. And so like through this research, I told you that we have a group that looks into, so my group looks into synapses and neuronal communication. How does it get affected in diseases, but also how does it get affected during learning? And then I told you about a zebrafish group. So we have a developmental biology group, which really looks into the sensory systems in zebrafish and what are the genetic factors and affect these mechanosensitivity. This is Agni Das Gupta. You guys will be hearing all about his work. And then we have Subhashish Ray, who's looking into one of his part is looking into the computational aspects of or doing computational modeling to match these different scales all the way from genes to the circuits. So he has a very cool computational model called as or simulator called as Moose. So you'll also hear about that. So yeah, I think that's it. We have a very vibrant team at Chinta. These are just the facilities. You guys will all be seeing this facility for your time here, I'm sure. And then we have people from all over. So I think one of the cool things to do all these cool things, you also need cool people. And I think Shona did one thing good. He was able to attract some really good crowd in the beginning, and people with international exposure. They were able to come to this city of Kolkata. So yeah, with that, I would just like to welcome you all. And now I can tell a little bit about maybe the summer lecture series. So again, it's going to follow the same philosophy. We thought we'll like give, I know this is the summer gap for you guys. You already are taking a lot of these courses, so you don't want to take more courses. But we thought, because a lot of you guys don't have neuroscience background, and to really appreciate the science that's being done here, it's important to understand the basis of it, right? So you can appreciate the images or the pictures even more. So we thought we'll just give you guys a sense of some basic neuroscience understanding, but through that, we'll also tell you what cutting edge research is happening in all these different domains of neuroscience, because this is not very common in India that you find an institute which is dedicated for neuroscience research. And so we'll have 7 more lectures, so 8 total lectures, including today's, where we'll cover these different domains. So we have Kritika, who will be talking about the cognitive aspects, the one that I talked about, the behavioral assessments that they use for these SCA12 patients and other movement disorders. So she'll give a lecture on that. Then I'll give a lecture on the basics of neuronal communication, the basics of synaptic transmissions, what synapses are, what are the different, why neurons are so beautiful, and also what's unique about neurons, why are they electrically excitable, and how do they learn? So what's the cellular basis of learning in memory? So I'll give 2 lectures on that. Not too much basics, but more about the cool science that's happening currently in this field. Then we'll have Subhashish Ray. He'll talk about the modeling approaches to really study some of these synaptic transmission and synaptic plasticity paradigms. And I also plan to talk about the olfaction a little bit. So yeah, so he'll talk about the computational aspects of neuroscience. He'll give 2 lectures on that, again, some of the newest things that's happening. And then Agnik is going to talk about developmental biology and zebrafish. Actually, not just zebrafish, but comparative biology, comparing how these mechanisms could be conserved across species.